well. Quick review. That first of all, eyes on the prize. Glow in the dark cat. That's where we're going. Thing one you need is easy. You just bust up the cells and extract the DNA. That's actually relatively easy. Thing two you need is DNA scissors, right? You need restriction enzymes, of which there are a bajillion. It just kind of depends on what sequence you want it to look for and how you want it to cut. Two kinds of cuts are a straight cut, which is fine for things like DNA fingerprinting and other applications. But if you want recombinant DNA, in other words, DNA that will recombine with other DNA, you want a crooked cut like EcoR1 does. Thing two you need after you cut up the DNA is DNA sorting. You need gel electrophoresis to sort the pieces by size. And it works because you're basically pouring a block of Jell-O and then injecting those mixed fragments at the top of the gel and using electricity to drag them through the gel, which sorts them because little pieces move faster than big pieces. Any questions so far? Those are the big two. All right. Eyes on the prize. So the process of taking a gene from one cell and putting it into another is called transformation. And I think I already told you about the first and simplest transformation that had medical applications, which was they took EcoR1 and cut open a bacterial loop of DNA called a plasmid and stuck in the gene for human insulin. Did I tell you that story already? Yeah. Good. And that's why Genentech is now worth billions of dollars, because they can now make insulin from a big vat of bacteria. And from there, they really took off, and they make everything now. You just got to get that human gene to combine into the bacterial DNA. There is some other, trickery you got to do. You got to make sure all the right promoters that allow, remember, RNA polymerase to bind and do the transcription stuff. There's stuff you got to do as well, but the basics is pretty simple. You've now got a bacterium that is expressing a human gene. So that's cool. So the third tool that you need to know about, and you don't need to know much about it, is called PCR, polymerase chain reaction. It's basically a way to multiply DNA. It's basically artificial DNA replication. It's like, hey, we got these pieces of DNA we're interested in, but we need a lot more of them to do any work with them. Put them in a tube with the right chemicals and put them in a PCR bath. And after a few hours, you will have millions of copies of each of those pieces. So that's what you need to know.
Don't they like PCR tests?
Yeah, very good. Absolutely. The COVID test, not the one that takes a few hours, but the one that takes a couple days, is, I believe, PCR based, where they the DNA products that get made from their test of you, they then amplify so that they can tell if they're there or not. Yeah, very good. It's actually a really interesting process. It involves, you remember DNA polymerase, right? The one that copies DNA when DNA replicates. Basically, somebody got the idea. Well, okay, we don't need helicase. We can fake helicase by heating and cooling the DNA. If you heat DNA enough, it splits. So we don't need the helicase. What we need is a polymerase that can survive the heat that splits the DNA. And this genius guy was like, let's take DNA polymerase from the bacteria that survive hot springs. There's bacteria that live at the bottom of hot springs. And so they took the DNA polymerase from those bacteria, and you just put that with your DNA in little tubes, and you literally put it in a water bath that heats and cools it periodically for like 2 hours, and suddenly you have more DNA. It's really cool. But you don't need to know all this either. The technology we've got, we can basically sequence your DNA by machine at this point. That's what 23andMe is about. Yeah. Yep, there are various companies that will do that for you with varying degrees of accuracy. It's an interesting thing to do, at least. It certainly started a lot of family arguments. No. But like, there's awful lot of white families in this country that have been claiming Native American blood for a long time that have recently learned that nope, not so much.
Like all German.
Yeah. Okay, so there's really just one tool left. I think there's a couple things left. Just a very simple application of gel electrophoresis and restriction enzymes is DNA fingerprinting. So it's worth saying what that is, how you do that. Basically, someone gets killed. I find blood at the crime scene. I think Brady did it. So, huh?
You just admitted to it. Don't even have to fingerprint.
It's the kind of thing Brady would say, when it was actually you and he's covering for you. So if I think that, what I gotta do is get Brady's cells. Doesn't have to be blood. Yeah, he doesn't really even have to want to give me the cells. Like, he leaves them everywhere. Just need like a drinking glass or anything with even a little bit of his DNA. What's cool is with PCR, I can make a whole bunch more, enough to analyze. Then I need some of the cells from the blood at the crime scene. And then I need some of the victim's cells because I want to make sure that the blood isn't from the victim, right? So I get all those cells, I shake them up with the right cocktail of restriction enzymes. And there's, pre-made restriction enzyme packs that are for this, that are just for DNA fingerprinting. You shake them up with that, it cuts them up into a bunch of pieces, but it cuts everybody's DNA slightly differently because you have about 0.1% difference between your DNA and the next person's. And you remember how the cone in the gel makes the different wells, and we were wondering why so many wells? Well, we're going to need one for the victim, one for Brady, one for the blood, and one for a set of standards, so we know everything's working right. There's 4 right there. Anyway, you run it, sort those pieces, stain the DNA, and then you compare where the bands are, right? Because the bands move a distance according to how many base pairs long they are. It's real easy to go, all right, all the bands match. It was Brady, or at least it puts Brady at the crime scene. It was bleeding from somewhere or other. More incriminating is when they can find someone's DNA, say, under the victim's fingernails, right? Signs of a struggle, that kind of thing. So that's the one of the forensic applications here. Questions about that? This is also how paternity testing works, right? To figure out if someone is a parent of someone else. They're not going to have the same bands, but their bands will be closer to each other, right? They will have more of the same bands than 2 people who are more distantly related. Let's not write that for now. Yeah, in fact, oh, yeah, so this is sort of a quick and dirty version of gel electrophoresis after, right? Because you cut it up and then you run it. And that's a good approximation of what the pattern of bands looks like on a DNA run. Yeah, don't write this, but know that every year more and more of the food you eat is made from plants and animals that are transgenic, that in other words have gotten genes inserted into them, like extra growth hormone genes to make them grow faster. Because that's the best thing for a farmer, right? Is if their animals or plants grow to adulthood as fast as possible. They can sell them for meat and then grow more. We've made mice with entire human immune systems, which is of course useful if you want to test how the human immune system reacts to something, and you can do it with a mouse. Pretty convenient. But yeah, like these are old numbers, and already, you know, this is probably 7 or 8 years ago, and already over half of soybeans were transgenic. Between corn and soybeans, that's most of what you eat, or most of what you ate. Well, if you eat beef, the beef ate corn, unless it's fancy grass fed. If you ate chicken, the chicken ate corn, and that corn was most likely transgenic. So, yeah. No, not inherently bad. And there are lots of different viewpoints on this, and I'm not saying that using as many transgenic organisms as we do is a good or bad thing. That's something you'll have to figure out what you think. There are lots of issues with ownership, because as soon as a huge company like, I think Monsanto is the biggest, as soon as they patent a genetically modified plant, they own that plant and its entire existence, basically. So, like, there's situations where farmers who normally can plant their crops and then use the seeds from their crops to plant new crops, that's illegal if you're using proprietary genetically modified corn, right? So there's ownership issues like that that can be real confusing and sometimes maybe unfair to farmers. So that's one side of the issue. Then there's the people who think there's health issues, and that may or may not be true. So it's a fairly complex issue.
So far, or like health issue wise, or
No, I have not seen, at least in the most commonly used genetically modified crops, I haven't seen reliable studies that say that just being genetically modified is inherently harmful. But that's, yeah, you know, like there may be stuff we don't know about. It's a very current issue. All right, so we're finally there. The thing you really need to know to finish the glow in the dark cat. You know how to extract, well, you know how to cut up the DNA, sort the DNA, find the gene you want, stick the gene from the firefly into the cat cell, but that's only one cat cell. Cloning, which is the taking of one cell and creating an entirely new organism, that's the last step. Does sexual reproduction normally produce clones?
no,
Why not?
because
What 2 reasons?
because you have 2 different sets of DNA going together, so it makes a new set of DNA.
Beyond that, it's not just that. It's not just that there's 2 parents. Who knows the answer I'm looking for that I've been harping on for quite a while now? What are the 2 things that make sure that sexual reproduction does not produce clones? Because even 2 parents combining their DNA, shouldn't that be able to happen the same way twice?
Twins.
That's not how twins happen.
The 2 DNA mix. I forget what the exact name was, but we talked about it in meiosis.
Yes, we did. It is in meiosis. Come on.
Like,
Come on, somebody's got it. 2 things. One happens in late prophase 1, one happens in metaphase 1. You got it? No.
Oh, it's that thing where we they like mix up and then they go together.
Yeah!
And they mix up
What is that called? Crossing over.
Oh.
Ding, ding, ding. And independent assortment. Remember? Remember? I had to, yeah, you had to write that. Okay, cool. But every once in a while, more often now than before, we get clones in humans. But first of all, let's talk plants. That's really easy. You can just, most plants, there's parts you can take from them and grow a whole new plant. You know that, right? Some plants you can just take a leaf and put it in water and it grows roots. Crazy. Houseplants do that. And that's going to be genetically identical to the other plant. That's cloning. Low-tech cloning. Animal clones, though, since they all reproduce sexually, doesn't happen much. The exception is identical twins. The way that happens is they do sexual reproduction to produce one cell, and then that one cell does mitosis one time. And you get two initial cells with the same DNA, and each one develops into a baby. That's not the same as fraternal twins, which is 2 sperm, 2 eggs. Totally different. Not clones. I guess identical twins aren't. You probably won't. You don't want to call them clones because they probably wouldn't like that. You're clones. But the kind of cloning we're thinking of here, really, there's not a whole lot left. Like it's to the point where we can clone pretty much anything we want.
The last time I heard about cloning, it was like last year. They were like, "Oh, we cloned a sheep," and I was like, "Oh, that's cool." And now we can clone anything.
Dolly, you're talking about?
Yes.
The first mammal to be cloned last year, you think?
Well, I heard about it last year.
Okay, well, let me just look it up because I don't think you were born yet.
Nope.
No. There are issues with lifespan at times. I don't know exactly what they are. 1996. Yeah, 1996 was when Dolly was cloned.
So now they can.
R.I.P. That's how you clone a tree. You just got to scrape off some root tip cells, and then you can culture them and grow a whole new tree. But yeah, she's the story.
Oh my goodness.
Dolly was the first one. A little too on the nose. A Scottish guy named Ian Wilmot was the one to clone Dolly.
Which one's Dolly?
Dolly's the white-faced sheep. The other sheep there is actually the sheep that gave birth to Dolly. Notice I didn't say Dolly's mother.
Oh.
Here's how that works.
No.
Because it's time, right? We're shooting for glow-in-the-dark cat here. Here's how they did it. They took the egg, ovum, egg cell, from the sheep that they knew was going to be the one that gave birth to Dolly. And they sucked out the nucleus from it, which takes a very small needle. You suck out the nucleus. And then they took one cell from a white-faced sheep, and they shocked the two to get the empty egg cell to fuse with the white-faced sheep cell. Then they got the right mix of chemicals to basically trick the egg into thinking it was fertilized. The egg developed into an embryo, and then they implanted the embryo into that original black-faced sheep. And then waited however long it takes to get a sheep.
And then they got a sheep.
Sheep gestation period. I don't know, 2 months? Something like that. Doesn't matter. And then they got a white-faced sheep genetically identical to this one. But like, are you my mother? Interesting set of questions that comes up when it's not a sheep anymore. Let's say I wanted another Caleb.
No.
I was really attached to Caleb and he was getting old.
I'm about to die.
Listen, it would not be that hard. I got to get a cell from Caleb. And again, it's not really up to him to give me one. I could get cells of his if I wanted. A glass, piece of his hair, right? And I do exactly this. I get a female human crazy enough to donate an egg cell, get the nucleus out of that egg cell, take the DNA from his cell and put it into the egg cell, or shock them to fuse them. There's sort of two different ways to go about it. Get it, sort of trick it into developing an embryo, implant the embryo into that original woman, and wait 9 months. What do I get? Is it Caleb? No, it is not Caleb. It is Caleb's identical twin, 16? 16 years younger.
Caleb with a 6
year delay.
Interesting.
It's,
So, okay, here's the steps.
it's
It's
Dolly named after Dolly Parton.
I don't know.
The only thing was Dolly. Dolly's wrong though. Dolly.
Dolly Madison? I think she makes cakes. Okay, so the steps here, because you need to know, if you're going to do the cat thing, you need a cell from the mammal. That cell needs to have a full set of DNA. You need an egg cell from the same species. This is where cloning dinosaurs gets hard. Because you don't have any living female dinosaurs to do this part. What about, like, what?
I mean, aren't they?
Similar.
Rhinos are mammals. Closest living relatives of dinosaurs are birds. So they're probably the best bet. That's kind of how they got around it in Jurassic Park. Yes?
Working in cloning mammoths with like elephants.
And it has to be something that's fairly genetically close. So the word for sucking out the nucleus is enucleate. You enucleate the egg. That sounds very evil genius, doesn't it? Then I enucleate the egg. Of course. The first fairly tricky part of this that is going to be the part where you want to make sure you got a bunch of Caleb cells, because you're going to screw it up a few times.
Oh God.
Oh,
You gotta either shock the empty egg and the Caleb cell to get them to fuse, or actually suck the nucleus out of Caleb's cell and inject it into the egg cell. Very delicate work either way. There's going to be some trial and error. But if it works, then you're on to the next tricky part, which is you got to get that cell to actually develop into an embryo. And that involves kind of being able to put it in the same sort of environment of hormones and chemicals that it would be in. Of course. All you need is one cell with intact DNA. So not only can you clone someone who's already died, but they can be dead for a pretty long time and still have those intact DNA cells. Abby 6.
Why haven't we done it?
Because it's not.
I don't know that we haven't. You know what I mean?
Yeah.
I know that it hasn't been publicly done, but who knows how much of this stuff has been done without anyone knowing? I don't know. But my answer, I guess, would be why would we do it? Which is your homework today is you get to dig into something. What we can do, what should we do? You know, where's your line? That's today's homework. Once this part's done, it's easy. You just, wait. You implant, and then you wait. And you get Good. Well, okay, we got a little baby newborn, Caleb's identical twin. Who are the parents? Is the woman who gave birth to him?
No.
She's gonna feel that way. It's a pretty strong attachment that happens when
Wait,
you
wouldn't it be?
spend 9 months carrying a baby and give birth to it. How about the company that sponsored the whole thing? Do they own the little baby Caleb?
Isn't that slavery?
Right, I think we're done with owning people, aren't we? I'm hoping. Yeah.
If you clone Caleb, would that make Caleb like the dad or its brother?
Right. See, that's the question. What if Caleb didn't want to be cloned? What if I just got his cells from the lip of a glass he was drinking out of?
You wanted, you were the one that was like, I'm gonna clone this.
So am I responsible now?
Yes.
I don't think so. I'm just the money guy. I'm just a, like there's all kinds of weird issues that come up.
Isn't it illegal to like clone somebody?
Oh, I'm sure. But are there really laws that specific? Have we made those laws yet? Is the question. That's what has scientists like meeting and stressing out over like, hey, we can do all this stuff now. We got to figure out what's allowed and what's not. It's important. So basically, your homework looks like this. Don't write this down. What I have is I did my best to create sort of a spectrum of least controversial to most controversial. You're going to get it digitally, and you got to color code it. Basically, green, yellow, orange, red. Green is you think it should be allowed. Red is totally unethical. Yellow is like some cases, but not others. Orange is like only very special cases should it be allowed. Okay? So read through for now. I'm going to post it in Classroom, so you'll need a device.